Benefits of intermittent fasting. Intermittent fasting has rapidly gained popularity as a dietary choice to help with weight loss and improve metabolic health. This dietary approach does not dictate ingredients or portion sizes, but delineates when and how long to eat. Intermittent fasting has been described as a general term for the various meal timing arrangements that cycle between voluntary fasting and non-fasting. Anthropologists believe early humans evolved to function in tandem with this scarcity and abundance of food cycles. They argue that how the body is nourished may be as crucial as the tonic. The concept of intermittent fasting has been incorporated as dietetic practice in many religions and cultures around the globe. This aims to clarify misconceptions and showcase relevant evidence for the various health benefits of intermittent fasting. Indeed, numerous claims about this diet have often been repeated without looking at the primary source. Here we give an overview of the main applicative benefits. Our clinical message is that intermittent fasting is a lifestyle intervention that requires well-organized planning and a flexible planning vision that reframes moderate caloric intake within a construct of healthier lifestyle moves. We will not generally restrict or address the physiology of time-restricted eating. As with healthy dietary patterns, benefits should be explored through a mixture of macronutrient composition and calorie reduction. Overview of intermittent fasting. Intermittent fasting is an eating pattern that cycles between periods of fasting and eating. It doesn't specify which foods you should eat, but rather when you should eat them. In this respect, it is not a diet in the conventional sense, but more accurately described as an eating pattern. There are several different intermittent fasting methods, all of which split the day or week into eating periods and fasting periods. Arguably, the most common form of intermittent fasting is the 16/8 method. This includes the feeding window, which has an 8-hour window during which to eat, and non-eating fasting time of 16 hours. There's also the 5-2 diet, which involves significantly reducing your calorie intake twice a week and eating normally the rest of the time. Intermittent fasting can have many benefits and is more effective than regular dieting because it can help you hold on to more muscle. When we fast, the body uses stored sugars in the liver and glycogen in muscles to fuel energy needs. Once that stage is completed, the liver starts burning fat and converts some into ketone bodies, which are transported to the brain. This is part of the stress response triggered by a lack of food. Some studies have investigated how increased stress like this may strengthen cellular defenses and protect against age-related diseases. This is the theory behind health claims for fasting diets, but more research is needed to confirm the effect in humans. Cellular benefits. Intermittent fasting gives cells time to rest and rebuild. Although it sounds drastic, fasting triggers a process known as autophagy, which is the body's way of cleaning out damaged cells and regenerating new ones. Autophagy is a fundamental process for cellular health. During this state, cells degrade and recycle components in a balanced and controlled manner, which enhances repair processes. Several studies suggest fasting may promote improved cellular function and longer life in aging organisms. The cellular stress response mechanisms activated during fasting can enhance the body's resilience against various diseases, benefiting overall health and cellular aging. For example, fasting has been found to improve metabolic health and reduce the risk of chronic disorders such as diabetes, obesity, and heart disease. Furthermore, research in animals suggests a role in protecting against age-related diseases such as cancer, which could be due to the enhanced body-wide maintenance and regenerative potential provided by autophagy. This is then reflected at a cellular level as studies suggest cells function better and deteriorate more slowly during intermittent fasting. Although more human research is needed to establish a full link to age-related diseases, intermittent fasting provides increased cellular rejuvenation that may benefit overall health for much longer. Autophagy and cellular repair. One of the key components of cellular repair is autophagy, a natural process within cells that removes damaged or non-functioning components and makes way for new components to maintain function and health. Nutrient and energy sensors within the cell mediate the activation of autophagy in response to nutrient scarcity. Emerging evidence indicates that intermittent fasting is a potent trigger of autophagy, increasing the natural process of autophagy and allowing for more efficient repair mechanisms depending on its steady-state level. Fasting length correlates strongly with a rise in autophagy in humans, with a 48-hour fast almost doubling the levels of autophagy compared to a 24-hour fast. Indeed, longer fasting durations would thus increase autophagic load, reducing mTOR activity and enhancing autophagosome formation, maturation, and lysosomal fusion mechanisms. Animal studies reveal that varying fasting protocols can enhance tissue-specific levels of autophagy, such as a 24-hour fasting regimen in mouse models, increasing levels of autophagy within the hippocampus but not affecting fasting-induced autophagy in the liver. In both rodent and human models, it has been shown that calorie restriction increases autophagy and reduces inflammation. As calorie restriction is an established activator of fasting-free autophagy, it is plausible that fasting-induced autophagy may also dampen key markers in inflammation, such as a decrease in IL-6 or TNF-A. In rodents and C. elegans, intermittent fasting is linked to a prolonged lifespan. It can help delay the onset of conditions or diseases such as cancer, metabolic pathology, or neurological diseases. Given that fasting-mediated benefits include longevity, aging, and inflammation reduction, the level of autophagy achieved for maximal beneficial effects would likely have to be reached consistently through regular fasting or long-term shifts in nutrition. Metabolic benefits. When an individual fasts, they do not eat for a prolonged period. This leads to many metabolic changes that vary with the length of fasting and from person to person. These changes include increased fat oxidation, liver glycogen breakdown, and the metabolism of circulating amino acids to help organs function properly, so-called glucose production. Additionally, tissue energy utilization increases while others, like the muscle, decrease. These changes allow the body to use different energy sources and optimize energy utilization to ensure essential functions of the body stay running. Interpreting these physiological changes and many others on a clinical basis is difficult. Fasting helps to improve insulin sensitivity. This is how efficiently someone can use insulin to signal to the body that nutrients need to be picked up and metabolized. The opposite, insulin resistance, is when higher insulin levels are required to have the same effect, lower insulin concentration in a sensitive individual. A study in adolescents showed a decrease after 2 weeks, 3 months, and 6 months, all with a fasting protocol. When fasting, individuals eating with a pattern also present lower blood sugar than those with another pattern. Furthermore, after 4 weeks of alternate day fasting in a group of middle-aged people, the blood value of bad cholesterol decreased. The body also contains lower triglycerides, while waist circumference decreases. Another parameter characterizing the metabolic improvements of intermittent fasting is reducing fat-free mass, subcutaneous and abdominal fat, and body weight. A study showed that people can lose fat-free mass after constraints and strength training. Improving insulin sensitivity. Insulin sensitivity is a significant component of metabolic health. It refers to how effectively a given tissue or cell insulin. In most cases, insulin sensitivity is discussed about glucose metabolism since it consists of the principal role of insulin, which is to control blood sugar levels. Insulin stimulates the uptake of blood glucose into muscle tissue for fuel and storage, thereby regulating overall energy metabolism. Furthermore, insulin inhibits hepatic glucose production and promotes hepatic lipogenesis through direct effects on the liver. The secretion of insulin and insulin action are tightly linked since the body secretes insulin in response to nutrient consumption as a media that should allow tissue to take up the blood nutrients, especially glucose, reducing blood levels of such nutrients. Different nutrient ingestion and fasting periods virtually affect insulin secretion and action by physiological and cellular mechanisms. Over the last several years, many studies have examined how insulin secretion and insulin action are affected by different periods of nutrient ingestion and fasting. Human studies have shown that short-term fasting increases insulin-mediated glucose uptake, enhances peripheral and hepatic insulin sensitivity, and reduces hepatic glucose production in healthy individuals while reducing basal insulin secretion and improving cellular function in individuals with abdominal obesity or type 2 diabetes. Consistent with these results, endocrine and cellular adaptations have been documented in skeletal muscle, adipose tissue, and liver to enhance fat mobilization and increase the use of alternate fuels during fasting. Longevity effects. There is increasing interest in whether lifestyle interventions such as intermittent fasting can benefit longevity. The term longevity comprises two concepts: lifespan, or the length of an organism's life, and health span, or the number of years lived in a healthy, disease-free state. Extending lifespan is currently an area of great interest to researchers studying aging as a biological process. Increasing health span also seems attractive and could bring significant social and economic gains. Findings from preclinical models show that when foods are restricted, like intermittent fasting, they result in a substantial lifespan extension across diverse species. Some effects of caloric restriction and weight loss on aging and wellness could occur by improving how cells and tissues function. The activation of several metabolic and cellular signaling pathways, triggered by reduced glucose availability, may mediate the benefits that fasting in its various forms appears to have in reprogramming cellular or mitochondrial efficiency. In addition, like caloric restriction, fasting also kickstarts cellular stress responses. This results in a mild cellular stress resistance that manifests, for example, as positive effects on cardiovascular health. Another way intermittent fasting helps reduce the likelihood of age-related diseases is through its ability to correct physical and metabolic parameters that, in turn, improve overall cardiovascular health and potentially metabolic health previously damaged by free radicals or reactive oxygen species. In essence, fasting might be a good strategy to shore up the cell's defenses to promote more extended health. Fasting plays a role in promoting resilience in the face of a set of changes that are physiological regulators of the aging longevity transition. Evolution has selected mechanisms such as apoptosis, cellular senescence, or autophagy to control the length of life of an organism. In particular, apoptosis and senescence avoid the spread of harmful agents. However, the former has a pro-inflammatory outcome, while the latter has noxious agent interactions, eliciting an inflammatory response by the immune system. Fasting might between these two cellular programs in a manner that delays aging and extends longevity. Our baseline inflammatory markers may further support the translational role of fasting for human health, since they were decreased in response to fasting, which is potentially beneficial from an anti-inflammatory and diabetogenic perspective. Study participants engaging in continuous fasting had significantly reduced inflammation during the fasting intervention, but returned to baseline values. Although transient, these findings still suggest that the acute increase in inflammation that results from fasting is counterintuitively protective. In our study, these results indicate that fasting is practical, effective, and read. It is an available lifestyle intervention for longevity that preserves or promotes health in older individuals. Impact on lifespan. Intermittent fasting can dramatically affect the rate of aging and maximal lifespan. It has been shown that intermittent fasting can suppress various parameters of aging, reduce the incidence of numerous age-related disorders such as type 2 diabetes, cardiovascular disease, neurodegenerative diseases such as Alzheimer's and Parkinson's disease, and enhance functioning in a variety of mental tasks. At the molecular level, intermittent fasting has been shown to trigger sophisticated hormonal alterations throughout the body. The net result of fasting resembles the idea of caloric restriction, as both induce blocks to various pathological states. Intermittent fasting can trigger a specific set of metabolic changes, inflammatory reactions, and micromechanisms. There is evidence in human tissues and mixed human evidence from epidemiological studies that regular fasting can extend human lifespan, particularly when coupled with a reduced carbohydrate diet. Limited human clinical reports have shown the positive effects of intermittent fasting consumption regimens on pathological aging and human mortality. However, a consensus is yet to be reached. Practically, the difficulty of aging research is that the genetic variability between any two individuals can be so vast, making it difficult to establish an effect nature of any given intervention in a short lifespan. Nonetheless, the evidence is indeed most promising. When it comes to fasting and fading metabolic memory of overweight conditions, periodically shifting between periods of restricted dietary intake can slow down or even prevent the neurodegeneration at the cellular and genetic levels that is characteristic of Alzheimer's disease. In addition, specific genetic pathways influenced by fasting are key for slowing aging. This information gives clues for developing drugs that mimic these fasting-induced longevity signals. Mitochondria play an essential role in how people age. Generally, once someone starts to put on pounds, their mitochondria manage it in a certain way, and those decisions establish their future health. However, a study of people who went on reduced caloric periodic fasting or a very low carbohydrate diet showed that the mitochondria in these participants aged differently in a way that appeared to sustain their health for longer. These results suggest that people have much more control over how and when they age than was previously appreciated. Blood biomarker data from over 2 million participants allows for developing a novel aging clock based on these data.